This conference will now be recorded.
Hello everyone. Can you
Hello.
hear me? Yes. Can
Yes.
you hear me?
Yes, we can hear you.
Thank you. Okay, let's wait for a few more minutes to remaining speakers to join. Hello. Hello. Well, since I hope everyone is here and we have a lot to cover, we really should begin today's webinar. And I really appreciate you all for attending today's virtual conference. A very good morning, good afternoon, and good evening to one and all present here virtually with me today. Let me introduce myself. My name is Elena Jones, Conference Secretary at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. For the next 5 minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also the further instructions. Coalesce Research Group LLC is very delighted to welcome you all the speakers, supporting associations, students, and delegates present here virtually with us today. With the aim mark the scientific excellence by facilitation conversations, sharing knowledge, Coalesce Research Group follows through the objective of setting standards in delivering excellence in the research among the researchers, academicians, and industries. Promulgating the research through conducting events and online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. Within a short duration, we have succeeded in creating a platform for developing a strong professional network with the people across the world from the respective fields that they haven't connected in a while. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposium, and workshops, providing the best delegate experience, excellence networking opportunities, conversations leading to innovations, learning new opportunities, lasting work relationships, and professionalization with the professionals around the global network. Just recently, we have also started our new platform for open access pre-reviewed scholarly literature where are the contents are freely available without any charge to the readers. We aim to share the new recent advances through scientific publications, which will help in minimizing the borders and contributing the use of research works of eminent authors, scientists, research scholars, students, etc., across a broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its subspecialties to promote the ideas which will guide the development and facilitate the use of scientific information and ensure its quality, and also will be a connecting link between researchers and scientific community. We are committed in ensuring that our journals and disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. As being the very new journals, we have coined up some interesting journals. Also, we have formed the editorial board, and we will be very glad if any of your today's attendees, as speakers or students or delegates, could extend our support by checking the journals and will be interested in publishing an inaugural issue of any of the journals according to the area of expertise. We will also send an email with all the details about the journal after the commencement of the webinar. Do take your time to check the website. Thank you. Finally, I would like to thank you all the attendees of participant of today's webinar entitled Fifth International Webinar on Plant Science and Molecular Biology, and hope you enjoyed the webinar of gain some knowledge from the sessions today. We also hope your participation will contribute to new research findings that will lead to new collaborations and research initiatives. Before we start with the session, I guess some of you will be the new to GoToMeetings app. Let me explain you the first important tools required for the presentation. On the GoToMeeting app webpage, you can find your 4 icons at the bottom of the center: mic, camera, screen, and end button. So coming to the mic button, click on the to mute or unmute. I request you all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. And coming to the camera button, you can use this button to share or and share the web camera. If you have the option to share the webcam, you can do it, and it is completely your choice. And screen button, you can use this button to share your screen. This is the main part during your presenter. After I call your name from my side, I will be making you the presenter, and then only I will give the permission to share your screen. Then only you'll be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. And the last button, that is end button, this is your fourth icon where you can use this to leave the session. On the top center, you can use the options to change your display settings. At the top right corner, you will have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For the settings, you can use the button to check for the settings for your speakers and microphone. So as mentioned, kindly remain muted during the presentation so that the presenters don't get distributed. For any questions, you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to please give us a brief introduction about yourself before starting with the session so that we can know about virtually. And let's move on to our sessions. The first presenter is Dr. Weng Xing Yu, China National Rice Research Institute from China. From the title, the OS ELF31, OL ELF34 complex regulators multiple targets to determine heading in rice. Hello, doctor. Are you ready with your presentation? Please click on the mic button. Hello, Dr. Wu. Are you ready with your presentation? Sorry, doctor, we can't hear you. Please click on the mic button. Hello, doctor. You can find the mic button at the bottom of the center. Please click on the mic button to unmute yourself. Hello, Dr. Wu. Can you hear me? Please click on the mic button or you can just use your headphones. Hello, Dr. Wu. Can you hear me? Am I audible to you? Please click on the mic button to unmute yourself. Am I audible to everyone?
Yes, we can hear you.
Okay, thank you.
Yes, we can hear you.
Yes, thank you. Dr. Wu, please click on the mic button to unmute yourself. Hello, Dr. Wu. Please once again log out and log in once again. I think you have some technical issues. Please log out and log in once again. Hello, Dr. Wu. Can you hear me? Please click on the mic button. Can you hear me? Hello, Dr. Yu, can you hear me? Sorry, I can't hear your voice. Yes, can you hear me? Please connect to your headphones. I think you have some technical problem. Please connect with another laptop or any other PC. Hello, Dr. Veronica. Can you able to present your talk?
Yes, I can. Okay.
Okay.
Hello, everybody.
Yes, hello. Now I'm going to make you the presenter. Yes, please click on the share button to share your screen.
Sure. Just a second. Can you please let me know if the screen is shared?
Sorry, we can't able to see your screen. Please. Okay.
Yes,
Yes, perfect. Yes.
I can see it.
Yeah,
Yes.
we can able to see your screen. Yeah. Please make it as
Uh,
a full screen.
yeah, it's full screen. Okay.
Yes, thank you.
Respected researcher, respected colleagues, uh, professors, it's a great pleasure to be here. My name is Veronika Stilkovska-Georgievska. I'm PhD student at Faculty of Pharmacy, University St. Kirill and Methodius in Skopje, Republic of North Macedonia. And besides me in the research team for this research topic were Professor Gyosha Stevkov, who is a full professor at the Faculty of Pharmacy at Institute of Pharmacognosy. He is also the Vice Dean for Science and International Cooperation. Then Professor Nikola Geshkovsky, who is associate professor at the Institute of Pharmaceutical Technology, and my colleague Anna Trajkovska, who is PhD student and also teaching assistant at the Faculty of Pharmacy. Another activities that I do besides being PhD student is being teaching assistant at the Faculty of Pharmacy at the Institute of Pharmacognosy. And also I'm analyst at the Center for Natural Products at Faculty of Pharmacy. So for today's topic, I will discuss the multivariate analysis of physical attributes of different cannabis strain seeds in authentication purposes. So in order to get to the point of our topic and objective of our topic, I will give a short introduction regarding this subject. So, as we all know, cannabis industry in the last decade is expanding. And in order to expand in the right way, many regulatory requirements have been made throughout the countries. In some countries, this plant is legalized only for medicinal use, and in other countries it's used for medicinal and for a recreational purpose. But in order to follow the latest regulatory requirements, the companies have to ensure that they have a consistent product and they ensure good quality control of the product. one of the problems that cannabis growers are facing is the historic prohibition of cannabis, which stunted the scientific research on its production, and it left growers to rely only on guides and online resources based heavily on anecdotal information. So they are facing lack of information regarding seed quality, seed certification, cultivation, and harvesting of the final product. Um, in order to be clear regarding the term cannabis, we all know that we usually hear the term cannabis strain, and we have a lot of strains on the market. But I have to point out that the term strain is, even though if it's mostly used in the cannabis industry, it's not strictly correct in the field of botany. So the term strain is actually used to refer to a cultivar of cannabis. So we will later explain what is cultivar of cannabis and what is the differences between variety and cultivar. Here you can see a European Union plant variety database for cannabis, and here you can see the American database for cannabis for hemp from the Association of Official Seed Certifying Agencies. So as you can see, in Europe, we have 81 entries, actually 81 registered varieties of cannabis. Why is this important? This is important because we know the origin of the cannabis plant. So variety is a taxonomic category which ranks below subspecies where present or species, and its members are differing from others of the same subspecies or species in minor but permanent or heritable characteristics. So basically all of the plants that are registered in this database, which is variety database, means that they have passed a lot of testings and to ensure the genetic purity and to ensure the variety of the species. So the difference between variety and cultivar is that cultivar actually is a type of plant that people have bred for desired traits, which are reproduced in each new generation by a method such as grafting, tissue culture, or carefully controlled seed production. So most cultivars arise from purposeful human manipulation, but some originate from wild plants that have distinctive characteristics. So we have to point out once again that strain can be is used actually, it's to refer to a cultivar. Basically the cultivars can be produced by crossbreeding two different varieties, commercial and wild type variety, or they can be produced by crossbreeding a variety with another cultivar. So as you can see, the variations are very high, and we have a lot of cannabis strains, or as I may say, cultivars, and that's where the job for cannabis growers tends to get more difficult. We have to point out also that there are regulations for seed certification and for regulations for registration of a variety of cannabis. So here on the slide, I have the European technical document for registration and for testing for registration of plant variety, and here is the web page from US Department of Agriculture, which gives guidelines for plant variety certificate of protection. So basically here we have a list of tests that should be done and throughout a few years. So in order to ensure that the strain that you are applying to register as a variety is actually well tested and it has reproducible results and characteristics. But these regulations are different from the seed regulations. Cannabis growers often face inconsistencies, or if I may say, not lack of information in the seed certificates. So basically, in some of the seed certificates, it is not stated which varietal purity is the strain, or to ensure clear information regarding the variety or the hybrid plant, but they often use information such as THC or CBD ratio, time of flowering, weeks of flowering, etc. So they are facing a lack of information, and it has happened before that cannabis growers buy seeds with seed certificates which have higher THC ratio, but when they end up cultivating them, they get plants with a higher CBD ratio, which is a very big regulatory problem, and they have to explain how this happens. Is this the problem in the seeds or a problem with cultivation? So that's why they need information such as genetic purity of the plant. Uh, this all starts with testing of the seeds. So the cannabis growers don't have much information regarding seed testing of the cannabis strains, so they can use the UPOV descriptors that I mentioned before from this technical document by the International Union for the Protection of New Varieties of Plants. But in this document, we have only one quantitative trait, which is 1,000 seeds weight, and two qualitative traits, which is the color of testa and seed marbling. So we did not see any physical characteristics of the seeds that are used as a distinctive characteristic for the seeds, so they can classify them or test them. At this point, our objective arose. So our objective is to examine the association between seeds' physical attributes characterization coming from different cannabis varieties. Our methodology was really simple, but in order to get to the methodology, we have to point out what kind of materials we used for our analysis. We collected seeds from wild growing varieties, as you can see. So this is the map of Republic of North Macedonia. It's in Macedonian. We chose two cities, Kočani and Strumica, in which we from these two locations, so as you can see, we have 8 other smaller locations at which we collected 1, 2, or 3 plants. So that means plants with seeds that were already produced in the time for seed producing period. So we separated the seeds from the plant, and we have 100 seeds per position. And we borrowed commercial strains for our analysis from some cannabis companies. They just gave us 100 seeds to scan them, and then we returned them back. We did not use them for further analysis. Why did we just borrow them? You will see on the next slide. And the methods, our methodology was very simple. We put the seeds on a scanner Epson Perfection V19, and we just scan them and save them as a TIF file, which is an image file. So this is a non-destructive method. It was very easy to use. So then when we scanned each image, we imported the image in high throughput phenotyping software for measuring seed size throughout image analysis. As you can see, this is the look of the software, and these are the processed seeds. It's very easy to use. It takes a little bit of time for processing because we have 100 seeds per image, but in the end the results are satisfying. We observed parameters such as area size, perimeter length, length width, and length to width ratio. This is how it looks, the data raw data exported from the software. The data is in millimeters and millimeters square. We have area size, perimeter, length, width, and length to width ratio. As you can see, these are all of the parameters for seeds for one location. in order to statistically evaluate the data, we had to sort the data per parameter. So as you can see, we did that in another Excel file. We sorted them out by length of seed, and these are all of the species, actually, the strains or varieties that we have collected. And we did that with the other parameters. And then we imported this data to SIMCA version 14.1 for multivariant analysis. We did principal component analysis, hierarchical cluster analysis, partial least square discriminatory analysis, in order to analyze the physical attributes, independent variables, and cluster affiliation dependent variables. Here are the results. The results are really clear. With the principal component analysis that was performed, was with two main components that cumulatively explained 99% of the variability of the parameters, the independent variables. Here is component 1 and component 2. And this is a very interesting point here, the loading plot, because it revealed evident relation among the parameters that describe the physical size of the seed. So here we have larger loading factors about length, width, perimeter, or surface area. But you can see that we do not have the same picture for morphology attributes. This is the length to width ratio, so which means that we really cannot distinguish the seeds if we want to use just this parameter. So this is not a very good indicator to show the differences between the seeds. To continue, we can see from the scatter plot that the first main component, which is connected to the parameters that describe the seed size, explains 78% of the variability of the data. And as I mentioned, the second component describes only 21% of the variability, and this is connected to the morphology of the seed. And as I mentioned on the previous slide, morphology of the seed, which means length to width ratio. On the basis that we knew where our seeds came from, and that means were they wild growing varieties or were they commercial varieties, we decided to do hierarchical data clustering, and was performed based on the parameters describing the seed size, and the affiliation of each sample to the two basic clustering group was further used as a discriminatory variable in the partial least square discriminatory analysis model. You can see here that with the clustering we have two major division groups. You can see that on the next slide that the statistical descriptors that we obtained with the partial least square discriminatory analysis model indicated towards satisfactory predictive capabilities regarding cannabis strain seeds classification based on the physical attributes. As you can see, we have two colorations here. So these are the two main groups of seeds. These are the commercial seeds. These are more dense, and here we have broader spectrum of wild growing varieties. So as you can see, these are more versatile and these are more compact. We have two main groups, but we have some subdivisions, as you can see here in the clustering, hierarchical data clustering. From all of the above mentioned, we can conclude that the sequential multivariate statistical modeling of the physical attributes data of various cannabis strain seeds resulted in development of partial least square discriminatory analysis model with adequate statistical indicators. So we believe that this model can be further used for prediction of cannabis strain seeds distinctiveness and uniformity based on its size-related physical attributes. So it will be informative to include this type of research in the technical documents, which are for varietal purity and for variety registration and testing. But this is not the end of our research. We have some future research directions that we want to take on. So it is to collect and include more data on physical attributes of different cannabis strain seeds. That means we want to collect different series of the same seed strains cannabis strains and from different locations, wild type varieties, and to analyze them in the same manner and to create a larger database so we can have more interesting data. We also have started with the chemical characterization of different cannabis strain seeds by different analytical techniques. The first technique that we already started is the tier analysis, which is a very interesting analytical technique, and we are in the process of collecting our data. In the end, we would like to make some correlation between physical and chemical attributes of different cannabis strain seeds, and if we can see and create a model for cannabis strain seeds distinctiveness, so the industry and the research community can use that model for further research. I would like to thank you for your attention. Yeah, if you have any questions later on, you can contact me on my emails, and I will be glad to answer to your questions. So once again, thank you for your attention and enjoy the rest of the conference.
Thank you, Dr. Veronica, for your presence and presentation. Is anyone having any queries? I hope no questions. Once again, thank you for your valuable
Okay.
presentation.
Thank you so
Thank
much.
you. So let's moving to on our next session by Dr. Weng Chen Liu from China National Price Institute from China. Hello, doctor. Hello, Dr. Liu. Can you hear me? Hello, doctor. Dr. Liu, please connect to the computer. Yes. Can you hear me, doctor? Speak out. Hello, doctor. Once go to your settings and please connect to the audio. Once go to your settings and please connect to the audio. In GoToMeetings app, please go to the settings and please connect to your audio. Hello, Dr. View, can you hear me? Okay, we can able to see your screen. Please click on the mic button. Hello, Dr. View. Please click on the mic button to unmute yourself. Hello, Dr. Box. Please connect to your audio device from GoToMeetings app settings. Hello, Dr. View. Please kindly join from your mobile device so that you can able to talk.
Hello,
Yes, we can hear your voice.
can you hear me?
Yes, we can able to hear you. Please continue with your talk.
Okay, can I give my presentation?
Yes, you can. You can start with your presentation. Please make it as a full screen.
Okay,
Okay, okay. Please continue.
okay, wait for me. And good morning, ladies and gentlemen. I'm from China National Rice Research Institute. Thank you, organizing community for giving me this opportunity to introduce my work. The topic I will report to you today is OS-ELF4, OS-ELF3, and OS LUX HD1 GHD7 model determine handling in rice. The morning-phased LHY and CCA1 express the peaks at dawn, activates the daytime expressed genes PR9 and PR7, which together with PR5 replaces CCA1 and LHY. The PRS also replaces RVE8, which induces the expression of evening phase genes TOC1, LUX, and ELF4, ELF3. Oh, wait. Okay, wait. Okay, this is clearly ELF4 and EF3 with LUX forms evening complex, multiple proton complex that replaces the expression of day-phased genes PR7 and PR9. TOC1 also inhibits the expression of morning-phased CCA1, LHY, PR9, and PR7. Flowing time is a master output pathway of the circadian clock. In rice, OSGI is a critical clock gene. HD1 is an ortholog of Arabidopsis constans, which works downstream of OSGI and promotes handing on the short day, while delays handing on the long day by either ablating or replacing HD3A expression, which is conservative in the Arabidopsis GICOFT pathway. GHD7 is a crucial floral replacer on the long day. EHD1 is a critical floral inducer in rice. GHD7 suppresses EHD1 and HD3A on the long day. HD3 expression is induced due to GHD7 being too low on the short day. In addition, previous report showed that circadian genes are involved in regulating handing in rice, like ELF3 as a floral replacer in Arabidopsis. ELF3-1 promotes handing for replacing GHD7 on the long day. Also, several major circadian clock genes have been identified to regulate flowing in rice. How the circadian clock interacts with the volumetric flowing pathway to regulate rice handing remains to be elucidated. To identify new rice gene regulating handing, we screened an extremely late handing mutant ELH1 from our rice mutant library generated by EMS in Nippon Berry. The mutant displayed 63 days later handing than NIP on the NLD at Hangzhou, and 36 days later on the natural short day at Hainan. In addition, we grew the ELH1 mutant and NIP plants in controlled long day and short day. ELH1 was also showed extremely late handing. The leaf emergence rate of ELH1 had no differences with NIP, indicating that the delayed handing in the ELH1 mutant was due to prolonged floral transition, but not retarded growth on both long day and short day. We then performed cloning of the ELH1 mutant gene using the mute-map method. The mute-map analysis indicated a candidate region as end of chromosome 1, including 3 candidate SNPs, and the locus OS 01G 74020 encoding ortholog of Arabidopsis lax resulted in a stop gain mutation. Co-segregation analysis revealed these locals may be the candidate gene, and we also evaluated 2 other mutant alleles of these locals. To verify the function of these locals, genetic complementation assay was carried out, and the positive transgenetic plants were able to fully rescue the extremely late handing phenotypes on both NLD and NSD. When overexpression construct was transferred to the ELH1 mutant, the positive transgenetic plants expressed OS logs showed a normal handing phenotype. The OS-LAX mutants generated by the CRISPR-Cas9 exhibited a similar phenotype to the ELH1 mutant on both NLD and NSD, suggesting that the mutation in these locals is responsible for the extremely late hanging phenotype in ELH1. OS-LAX is an ortholog of Arabidopsis lax, and OS-LAX is highly homologous among both monocots and dicots species. OS-LAX was constitutively expressed among different tissues preferentially accumulated in leaf blades, which is called with a gas assay. So daily expression pattern analysis showed that OS-LAX transcripts were increased during the light period and reached a peak at dusk, and was reached to the lowest level at June on both LD and SD. However, the ratio and the amplitude of OS-LAX transcripts were slightly decreased when transferred to continuous light conditions, and declined sharply when shifted to continuous dark. Moreover, the expression waveform or amplitude of OS-LAX was slightly higher in ELH1 than NIP, probably due to the negative feedback regulation. The following is strictly regulated by the scaling ratio clock. We examine the ratio expression of clock genes in rice. The expression of OSPR 33, 37, 73, 95, and 59, and OSPR1 were upregulated in ELH1. In contrast, the expression levels and amplitude of OSCCA1 were significantly lower than NIP, indicated that OS-LAX is involved in meeting the scaling clock of rice. To evaluate how OS-LAX regulates the rice flowering pathway, the expression of flowering time regulated gene were also monitored in NIP and ELH1 mutants for RT-qPCR. We first examined two floral integrators, HD1 and EHD1. HD1 transcripts were upregulated in ELH1 compared with NIP. ELH1 transcripts were almost undetectable in ELH1, indicating that OS-LAX replaces HD1 and promotes EHD1 expression. HD3 and OS-MAZE14, which lie downstream of HD1 and EHD1, showed similar expression patterns to EHD1. OSGI and GHD7, which are upstream regulators of HD1 and EHD1, their transcripts significantly increased in ELH1 mutants, suggesting OS-LAX promotes handling by suppressing OSGI, HD1, and GHD7. To further investigate the molecular mechanism underlying OS-LAX regulates handling, we explored why rice forms an easy complex similarly to Arabidopsis. In rice, there were 3 orthologs of ELF4 and 2 orthologs of ELF3. We found that the C-terminal domain and the middle region of OS-ELF3 to 1 could mediate physical interaction with OS-LAX and OS-ELF4, and OS-LAX and OS-ELF4 failed to interact with each other. The interaction was also supported by pull-down and lock assay. These results confirm the ELF 3.1 and X bridge to allow the formation of the ternary complex. The nuclear localization of OS-EC complex is vital for the transcription regulation of target gene. A transitioned expression assay showed that the OS-LAX was located in both the cytosol and the nucleus. Consistent with previous report, we also conducted the subcellular localization of OS-ELF 3 to 1 and OS-ELF 4, and found that they only localized to the nucleus. To examine whether OSEF3 to 1 affects the translocation of OS-LUX to the nucleus, we transiently co-expressed OS-LUX and OSEF3 to 1 in rice protoplasts. The fusion signal was detected only in the nucleus but not in the cytoplasm, whereas OS-LUX alone was localized to both the cytoplasm and the nucleus, shows that OSEF3 to 1 alters the subcellular localization of OS-LUX and promotes OS-LUX translocation into the nucleus. BIFC assay was conducted to detect the subcellular localization of the OS-LUX OSEF3 to 1 to OSEF4 interaction, and the fluorescence signals were observed only in the nuclear when expressing OS-LUX OSEF4 due to the existence of ELF3 to 1. OS-LUX failed to interact with OSEF4, which triggered us to consider the effects of OSEF4 on OS-LUX OSEF3 to 1 interaction. All results found OSEF3 to 1 contributes to the translocation of OS-LUX to the nucleus, so we want to know if OSEF4 might promote the interaction between OS-LUX and OSEF3 to 1. We used LCA assay to test this hypothesis. The relative luciferase activity observed for the OSEF3 to 1 OS-LUX interaction was significantly increased when OSEF4 were co-expressed with OSEF3 to 1 and OS-LUX. OSEF4 might promote the interaction of OSEF3 to 1 with OS-LUX. Thus, taking the results from co-localization, BIFC and LCA assay together, we conclude that the localization of the OS-EC complex in the nucleus and complete EC activity is necessary for result regulating output genes. To verify the genetic regulation relationship between the OS-LUX OSEF3 to 1 to OSEF4 trimer components, CRISPR-Cas9 was used to generate non-mutants for each of the 3 genes in the NIP background. OSEF3 to 1 mutant showed a 22-day and 18 days later handing than NIP under NLD condition and NSD conditions on the Hangzhou. LUX OSEF3 to 1 flowered as extremely late as OS-LUX. OSEF4 to 1, 4 to 2 resulted in late handing in Hangzhou NLD conditions. But OSEF4 to 1 and OSEF4 to 3 has no phenotype under NLD conditions, and OSEF4 to 1, 4 to 2, and 4 to 3 showed 5-day late handing compared to NIP under CSD conditions. The phenotype of OSEF3 to 1, OSEF4 to 2 was similar to OSEF3 to 1 under NLD conditions. Thus, our result suggested that OSEF4 may function together to form a ternary complex with OSEF3 to 1 and OS-LUX to regulate photophilic flowering. HD1 and GHD7 have been reported as crucial regulators of photophilic flowering in rice. The expression levels of HD1 and GHD7 were upregulated in ELFH1. In Arabidopsis, LAX binds to the LBS motifs. We found LBS motifs in HD1 and GHD7 promoter, which implied HD1 and GHD7 might be the direct target gene of OS-LAX. TRPQ-PCR analysis found a significant enrichment on OS-LAX on the S3 region instead of S1 and S2. The EMSA assay further confirms this result. LAX transitioned transcriptional activity assay showed that OS-LAX acts as a transcription repressor to bind to the to suppress HD1 activity. Similarly, OS-LAX also direct binds to the LBS in the GHD7 promoter to suppress its expression. To determine the effects of OS-EC complex on transcription repression of HD1 and GHD7, we performed a LAX transition transcription activity assay. When a OS-LAX of expression construct was co-transferred with HD1 LAC, the luciferase was significantly suppressed compared with the control, indicating that OS-LAX acts as a transcription repressor to bind to suppress HD1 activity. When a OS-ELF321 construct was added, the luciferase was reduced, showing that the OS-ELF321 to OS-LAX dimer complex could enhance the suppressive activities of OS-LAX to HD1. Moreover, when a OS-ELF4 were further added, we found that the luciferase was further reduced, suggesting that OS-ELF4, OS-ELF321, and OS-LAX complex has the most potent suppressing effect on HD1. There is a parallel case that the OS-EC complex has a similar suppressing effect on GHD7. Taken together, these results indicated that the OS-EC complex direct binds to HD1 and GHD7 promoters and suppresses their expression. GHD7 is a key floral repressor that forms a dimer complex with HD1 to suppress the expression of EHD1 and HD3A during light period to delay handing on the long day. HD1 promotes handing on the short day due to the low abundance of GHD7 to generate HD1 GHD7 repressive complex, and a similar pattern was observed in GHD7 and DTH8 mutant on the long day. So expression levels of HD1 and GHD7 were significantly upregulated in ELH1 on both short day and long day, means more HD1 and GHD7 proteins may be accumulated in ELH1. Thus, HD1 GHD7 complexes play a key role in determining delay handing in ELH1. This is why the lower EHD1 and HD3A in ELH1 on the short day are similar to those on the long day. There we gave a model about our theory. In wild-type, OS-ELF321 drag OS-LAX into the nucleus and formed OS-EC complex. The OS-EC complex binds to the promoter of HD1 and GHD7. The downregulated HD1 and GHD7 further realized the suppression of EHD1 and HD3A, then caused an early handing in the long day. On the short day conditions, HD1 promotes flowing in the background of a low level of GHD7, thus regulates the normal handing in rice. But in mutation of OS-ELF4 and OS-ELF321 weakens the repression of OS-EC to regulate HD1 and GHD7. In OS-ELF4 mutant, OS-ELF4 failed to improve the interaction between OS-LUX and OS-ELF 3-1. While OS-LUX, OS-ELF 3-1 dimer still plays a predominant role in target repression. Thus, a minor effect on handing in OS-ELF4 was observed, which delayed handing by about 5 days. For OS-ELF 3-1, the repression strength of OS-EC is significantly weakened due to OS-LUX being unable to enter the nucleus completely. Thus, the mutation in OS-ELF 3-1 caused a more significantly flowing effect. Defect OS-LUX failed to form OS-EC complex, resulted in the suppression function of OS-EC complex being entirely lost, which entirely released the targeted repression of HD1 and GHD7. Thus, an extremely late handing phenotype was observed in the ELH1. Thus, the complete OS-EC complex is required to control rise handing for repressed HD1 and GHD7 express. We are grateful to thank Professor Shihua Chen, Professor Li Yongcao, Professor Dr. Peng Xu, Professor Yingxin Zhang, Dr. Xiaxia Wen, Dr. Qingqing Yang, Dr. Lin Liu. And also thank the National Natural Science Foundation of China and the State Key Library of Rice Biology. Thank you.
Thank you, Doctor, for your presence and presentation. Is anyone having any queries? I hope no questions. So once again, thank you, Dr. Wu, for your presence and presentation.
Okay, thank you. And Dr. Chao, because he is not feeling good, he has a cold. So sorry, he can't give a presentation.
Yes, Doctor, we have received an email that he is not feeling well. Thank you for the information.
Thank you.
Okay, let's move on to our next session. Due to some technical issues, some of the speakers are not available right now. So I would like to call upon Dr. Georgia Willoughby from School of Sciences and Engineering, University of Nicosia, Cyprus. Hello, Dr. Georgia. Are you ready with your presentation? Hello, Dr. Georgia Willoughby. Hello, Dr. Georgia Willoughby. Can you hear me? I think she is not available right now. So I would like to call upon Dr. Ziliana Fedorovic from University of Split, Croatia. Hello, Dr. Ziliana. Hello, Dr. Fedorovic.
Hello, can you hear me?
Yes, Doctor. Can you hear me?
Yes.
Sorry, we are run up ahead of your schedule because some of the speakers are having technical issues. So
No
they
problem.
join later. So are you ready with your presentation?
Yes, I can upload it.
Okay, thank you. So now I'm making you the presenter. Please click on the screen button, share your screen.
Okay, can you see this?
Yes, doctor, we can able to see your screen. Please continue.
Okay, I will definitely first introduce myself. So I'm Željana Predotović from University of Split, Faculty of Science, Department of Biology. So I will talk about my study, phytochemical characterization and screening of antioxidant, antimicrobial, and antiproliferative properties of Allium cornutum clementi and two varieties of Allium cepa peel extract. So I will just start with the introduction. So we all know that the onions are one of the most commonly grown vegetable crops. So the common onion and the triploid onion, which is along with the common onion, often grown in the Dalmatia coast. So they are widely used, and they've been used for years in human nutrition. But also as the production and consumption increases, so does the generation of the waste materials from these different onion parts, raising the need for efficient ecological and ecological disposal and use of such waste materials. So in the EU, more than 500,000 tons of onion waste are generated annually. So these onion peels that are discarded during peeling have become a global ecological and economic problem. So for this very reason, scientists have decided to investigate the possible benefits of onion waste. There are different studies that have already shown that allium vegetables are really rich source of an important bioactive phytochemicals, especially sulfur and phenolic compounds. And surprisingly, the amount of these compounds is significantly higher in the waste parts of onion than in the eatable part of the onion. So I will focus on phenolic compounds present in the onion, which have shown significant biological activity from antioxidant to anti-cancer, antimicrobial activity. We must say that most of these studies conducted involved eatable parts, and very few studies were conducted with the onion waste. So that's why we try to investigate a little bit more this waste material or waste extracts. So considering all this, we first wanted to identify the major compounds in the methanolic peel extract of two traditionally grown onions in Dalmatia and to investigate their biological activity, antioxidant, antimicrobial, and antiproliferative activity. So I will start with the results because there is no space for the materials and methods. And if somebody wants to hear something more about some methods that we used, you can definitely contact me and I will send you the paper. So first, uh, we perform high performance liquid chromatography. So the chromatography analysis of these extracts revealed the presence of flavonols and anthocyanins in significantly higher amounts than the previously identified in onion bulb extracts. So there were five flavonols that were detected in all the extracts. So quercetin 3,4-diglucoside, quercetin 4-monoglucoside, and pure quercetin, isorhamnetin, and camphorol. The two major flavonols identified were definitely quercetin 3,4-diglucoside and quercetin 4-monoglucoside, the concentration of which was at least twice as high in the triploid onion Allium cornutum than in the other two common onion cultivars. The concentration of anthocyanins were significantly lower than the flavonols, so we definitely focus on the flavonols. Then we perform antioxidant analysis of these peel extracts, and the antioxidant activity was determined by two different methods, the DPPH and ORAC method. So since these extracts are really rich in phenolic compounds, which I just mentioned, and they are known for their ability to act as electron or hydrogen donors, they were definitely expected to have a significant antioxidant capacity. So the results showed that the antioxidant activity of onion extract was definitely correlated with phenolic content. As you can see at table 1, the extract of Allium cornutum showed significantly higher antioxidant activity than the red and yellow onion varieties in both assays at the concentration of 100 micrograms per milliliter, which can be explained by the higher concentration of phenolic compounds. So the second biological activity tested was the antimicrobial activity. We performed a broad dilution assay. So all the extracts showed better inhibitory activity on gram-positive bacteria as compared to the gram-negative. So again, the extract of Allium cornutum showed higher activity on all tested bacteria, yeast, and molds compared to the red and yellow onion varieties. The yellow variety and Allium cornutum were definitely showed definitely strong inhibition on growth of two Staphylococcus aureus strains. So with the minimum inhibitory concentration of just 7 micrograms per milliliter, which is really good at activity. Also, we can say that no antifungal activity of Allium cornutum or Allium cepa waste extract against Candida albicans was observed, which is definitely in agreement with some previously published data. Also, food poisoning mold, Aspergillus niger, was not affected by either red or yellow variety of common onion, and Allium cornutum showed really low activity against this fungus with a minimum inhibitory concentration of 500 micrograms per milliliter. The next, we perform analysis of antiproliferative activity of these extracts on 3 cancer cell lines: cervical cancer cell lines, colon cancer cell line, and osteosarcoma cell line. We perform MTS cell proliferation assay. So the cells were treated with various concentration of onion extracts and incubated for 48 hours. And then the half maximal inhibitory concentration value was determined. So in the contrast to the result of antioxidant and antimicrobial activity, in case of antiproliferative activity, the red variety of Allium cepa proved to be better than the yellow variety and Allium cornutum in inhibiting proliferation of 2 cancer cell lines, colon cancer cell lines, and osteosarcoma, as you can see on figure 1. Also, the Allium cornutum and yellow variety of Allium cepa showed similar and slightly better inhibitory activity on cervical cancer cell line. So we can say that the good antiproliferative activity of onions is certainly due in part to the quercetin glycosides, which have been identified as the major bioactive compounds of this extract. However, it is evident that there are other biologically active molecules that can also contribute to the antiproliferative activity. Also, we perform in vitro digestion of onion extracts since we can use it in food, probably in food industry. So we try to determine the stability of flavonols and the effect of on the antiproliferative activity. So you can see the figure 2. So the results show that these extracts remain quite stable after an oral and gastric digestion, as we found no significant difference in antiproliferative activity compared to the undigested extract. And in vitro small intestinal digestion, which you can see by the sign EF, so is definitely showed that and definitely increased the antiproliferative activity of the extracts on all cancer cells tested compared to the undigested extracts. So we might say that in vitro digestion definitely increased and bioactivity and bioavailability of this extract. So as a conclusion, we can say, first of all, that this was the first study of chemical composition and biological activity of peel extract from triploid onion, Allium cornutum, or we called it lutica. The results show that these peel extracts are rich in flavonols, which are the main antioxidant compounds, and they also possess good antioxidant, antimicrobial, and antiproliferative properties. So considering this large amount of peel waste that is generated annually, and which is an increasing environmental and economic problem, new studies should be done to see the possibility of their use as a source of biologically active phytochemicals that can be used in some part of the industry, especially pharmaceutical, cosmetic, or food industry. But we must perform new research, new studies to identify other biologically active compounds present in the wastes, and also the mechanism behind their biological activity, so we can possibly use them in some kind of industry. I think I was in time. So that's it.
Thank you, Doctor. Thank you for your presence and presentation. Is anyone having any queries?
Hi, hello.
Hello.
One question from my side is,
Yes.
yeah, can we use it with any combination of antibiotics, beta-lactam antibiotics, to reduce the growth of Staphylococcus aureus?
The Staphylococcus aureus, yes, they had the best effect on the MRSA, or clinical Staphylococcus aureus. So with the minimum inhibitory concentration of 7 micrograms per milliliter. So it was definitely the best effect. They could inhibit the growth of Staphylococcus aureus.
So it can be worked with the older beta-lactam antibiotics like as penicillin, ampicillin.
Yeah, I don't know. We should try, definitely, possibly, that it
Okay.
will have the better effect. Yes, but thank you for the suggestion. I think I will try that.
Yeah, actually 100 microgram per ml is I think higher concentration now.
I don't hear you. Just a second. Yeah, can you repeat the question?
Yeah, for clinical study, na 100 microgram per ml is higher concentration.
Yes. And this is the 7 micrograms per milliliter.
Yeah.
So
Yes.
it's quite
Yes.
good. It's quite good, but we must perform another experiment. And also, your suggestion is really, really good. So I will probably try it. Thank you.
Yeah. Welcome. Okay.
Thank you. Is anyone having any queries?
Yes, I would like to ask another question if possible. Um, have you performed any of these anti-proliferative tests on Allium sativum? Maybe have you tested Allium sativum, or it's just Allium cepa species?
Thank you for your question. So we didn't tested Allium sativum because it's really, I think, all of the world is tested Allium sativum, but also I choose Allium cepa because Allium cornutum, which I just mentioned, is the triploid species with 3 putative parental species. So Allium cepa is one of the parent species, Allium royle, and wild Asian species, Allium schemense, which are known for their good biological activities. So I really wanted to test probably also Allium cornutum and Allium cepa and compare it. I will try to find Allium royle species and Allium schemense and test it, and then we can say something more about Allium cornutum, but it's definitely better than the common onion, so we often use it here instead of the common onion. That's why I didn't take Allium sativum and just Allium cepa, because it is one of the parental species of Allium cornutum.
Okay, thank you for your question. It's very for your answer. It's very informative presentation, and the aspects, the anti-proliferative aspects are and activities are really interesting to hear, especially for colon cancer cell lines. Thank you.
Yeah, thank you very much.
Thank you for your presence and presentation, Dr. Zelena Ferovic. So let's move on to the next presentation by Dr. Ramdatta Bhatta from Tribhuvan University, Nepal, in the title from Production of profitability of hybrid rice is influenced by direct nutrient management practices. Hello, doctor. I think he left the session. Just wait for a few more minutes to connect. Hello, Dr. Ram Datta Bhatta. Are you ready with your presentation? Hello, Dr. Ram. Can you hear me?
Hello.
Yes, doctor, we can hear you. Are you ready with your presentation?
Yeah, I'm so sorry that I was out of network today. So may I present my presentation by tomorrow?
Okay, doctor. Okay, we can understand and we'll schedule your talk on tomorrow.
I'm so sorry that the network is not very good here.
Okay, thank you.
Thank you.
Thank you for waiting. Due to some technical issue, Dr. Georgia Willoki sent her video presentation. So I will now presenting her video. Please stay tuned. Thank you, George Jawlewski, for your presence and presentation. So I would like to inform you that due to some technical issues, some of the speakers are not available right now. So let's continue with the lunch. After we'll resume after 30 minutes. We will resume the sessions after 30 minutes. Please stay tuned.
This conference will now be recorded.
Hello, Dr. Thamraj H. Thamraj. So are you ready with your presentation? Can you hear me, doctor? Hello, doctor. Can you hear me? We can't hear your voice. Please click on the mic button to unmute yourself. Okay, in the meanwhile, let's move on to our next session by Dr. Tidra Hussain from Gomal University from Dera Ismail Khan, Pakistan. Hello, Dr. Tidra, are you ready with your presentation? Hello, Dr. Tidra, can you hear me? I think he is not available. Hello, Dr. Tamraj. Can you hear me?
Hello, Elena Jones.
Yeah, hello. Are you ready with your presentation?
I'm ready, yes, okay. Can I start my presentation?
Okay, so I will be sharing the screen. Is it okay, or you want to share the screen?
Okay. Şey buradan nereye? Screen elemeli.
Did you get the pop-up window? Yes, we can able to see your screen.
I could.
Yes, please open your PPT file.
Better. This is.
Okay. Hello, doctor. Can I share my screen?
First moment, please.
Okay.
This is my presentation.
Okay, okay. Yep. Yes, we can able to see your screen. Please continue.
Can I start?
Yes, doctor, you can start.
Hello, my dear conference. Thank you for taking the time to give me presentation at the webinar. I wish to start this is a part of the webinar. With your permission, I begin my presentation. First of all, let me introduce myself. I am Tamra Tamrazov, Doctor of Biological Science from the Republic of the Azerbaijan. Research Institute of Crop Husbandry, Department of Plant Physiology and Biotechnology. Particular attention should be paid to physiological study to increase resistance to drought and other stress factors of the wheat genotypes and identify sustainability mechanism on the other hand to determine the physiological and genetic change occurring in genotypes. The study of the effects of resistance on fertility measurements and the effect of fertility indicator on typical which investigated based on different soil and climatic conditions. In the 2020 and 2021 academic year, 90 different wheat genotypes should be measured in the caption experiments of the Institute of three groups, early, medium, and late maturing. As a result, two types from each group we compared including durum and bread, the net fertilization rate, stomatal condition, transpiration rate per leaf area, leaf temperature, and intercellular carbon monoxide concentration were measured using a portable photosynthesis system like on the flag leaves on midday, 9 o'clock and 20 o'clock after antithesis. During the study, we analyzed in the gas metabolism and some physiological features of different genotypes responding to drought stress. Such research will provide valuable information that can be used as a genetic basis for wheat production to increase productivity and productivity from stress. On the other hand, the difference between the variants in late-grown samples complete with the prolongation of maturation and the occurrence of acute drought. This also leads to loss of product. Purpose of the topic. The main purpose of the study interested from international nurseries and studies the dynamics of change in the main physiological parameters of durum wheat and bread wheat genotypes, diet to drought, adapted to local conditions, as well as to determine the impact of drought on the productivity of adapted genotypes. And condition of research. In the 2020 and 2021 academic year, 90 different wheat genotypes were measured in the Apcheron experimental base of the institute in three groups with varieties genotypes, early, medium, and late maturing. As a result, two genotypes from each group were compared, including durum and bread wheat. During the study, 90 samples of local wheat genotypes introduced from international selection nurseries and adapted to local conditions were studied. Out of 90 samples studied, more resistant genotypes were selected and placed in three groups. Despite the research was carried out on three groups in three regions, in the results of the analysis given in the article based on only one group. Genotypes of early ripening wheat varieties, genotypes of middle ripening wheat varieties, genotypes of late ripening white varieties. Observation and measuring in the field of experience. You are seeing grain yield and its components, grain yield, biomass, and number of spikes per 1 square meter for each cultivar were measured by harvesting 10 square meters, the central part of each pilot crop maturity. Harvest index was measured by dividing grain yield to mass production, the number of grain per spike, and 1,000 grain were measured on 10 randomly selection man shoots. In the first table, this table mainly shows the number of some spread and from which samples during the research, as can be seen from the table of 26 sample study and 18 four samples, 74 were red and 16 were durum wheat. As a result, 27 wheat samples were selected. 21 of them were bread and 6 were durum wheat. The monthly mean maximum and mean temperature, humidity, and precipitation and the site of experimenting 2001 years. This table provides information soil climatic condition of the country. The amount of precipitation for the individual months is given low, heat, and medium. At the same time, their humidity capacity was defined and given by months. This reflects the processes from sowing to harvesting to aquatic productivity, as can be seen from the stem. Special attention should be paid to irrigation condition, reserve water distribution rate of canal system, irrigation district, water distribution rate in the farmland, farmland transport rate, and photosynthesis rate, another dry biomass rate, harvest index, irrigation water distribution process, crop water use, and yield information processes. Grouping of study which starts by maturing. The dynamics, the studied with genotypes were evaluated and grouped according to the ripening period. It was mainly three groups of early growing, medium growing, and late growing. In the next dynamics, the studied types were evaluated and grouped according to the ripening period. It was mainly three groups of growing, medium growing, and late growing. Main comparisons of grain yield and its components and some morphological terms which require force and this. In this table, the rate composition of wheat and its components and some morphological features of wheat varieties in the following period can be seen from the table. Productivity by biomass productivity index were determined separately in the studied samples. In the next table, you can see in addition product indicator elements were analyzed and the results of analysis compared. It mainly calculates the weight of 1,000 tons, the number of grains and specks, number of specks of sugar meters, and finally stress resistance index. As can be seen from the table, most of the wheat genotypes resistant to stress. This table shows the correlation of the spike elements. Due to the correlation, there is a correlation between the weight of the grain spike and the number of grains in the spike. At the same time, there is a high correlation between the number of grains in the spike and the number of spikes. The lowest correlation was observed between the weight of the spike and the weight of the spike. Here, these images were taken during various morphological and physiological studies conditions during the growing seasons. Thank you for your attention.
Thank you, Doctor, for your presence and presentation. Is anyone having any queries? So I hope no queries. Once again for your presence. So let's moving on to your next session by Dr. Iqtidar Hussain from Gomal University, Dera, Ismail Khan, Pakistan. He is able to
Thank you.
present his talk today.
Thank you, ma'am.
Okay, so now I'm making you the presenter. Please share your screen. Please click on the screen
Wait
button to
some,
share your screen.
wait some time. I am trying to share my presentation.
Okay. Yes. We can able to see your screen.
No, you can see my presentation.
Yes, we can see your
My
presentation.
sound is clear.
Yes, clear.
Thank you, ma'am.
Please continue, speaker.
Bismillahirrahmanirrahim. I, Dr. Iqtidar Hussain, Assistant Professor, Gumal University, Department of Agronomy, Deir Khan, Khyber Pakhtunkhwa, Pakistan. My topic is advantage of biological nitrogen fixation in wheat-based legume polyculture in sustainable agriculture system. So comes to the climate change scenario in Pakistan. Agriculture systems are extremely vulnerable to climate change given their sensitivity to variation in temperature, precipitation, and occurrence of natural events and disaster. Meanwhile, emission from automobile and agriculture activities, many gases contribute considerably to global warming. Wheat, Wheat Triticum aestivum is an important member of Gramineae or Poaceae family. Wheat crop supports GDP in Pakistan with 1.7%, while 9.1% on account of value addition in agriculture sector and 8.7 million hectares are reserved for wheat cultivation in Pakistan, which produce about 25.5 million tons. Contribution of wheat during Rabi season cultivation is about 70%, covering 37% of the country's total harvested area. In Khyber Pakhtunkhwa province, from which I belong, it is cultivated on an average of about 48% area, covering 57% of the area under cereal crops. Continuous utilization of soil reduces fertility, therefore many physical and chemical changes happen in the texture of soil during a crop period. For this, a lot of dependence is done on synthesized fertilizer. This causes increase in cost of production and further change in physical and chemical properties of soil. Intercropping is a system in which two or more crops are usually planted together. Cereals are intercropped with legumes in a polyculture pattern. In this way, a land is effectively utilized and fertility of soil is also improved. With the objective of biological nitrogen fixation plus land equivalent ratio in mind, investigation were carried out to find the best wheat legume intercropping system with modern planting geometry techniques in the area. Biological nitrogen fixation Biological nitrogen fixation is the process that changes the inert nitrogen molecule into biological useful ammonia. This kind of nitrogen fixation is carried out by bacteria frequently found in the roots of nodules of leguminous plants. The most common bacteria is Rhizobium japonicum. Biological nitrogen fixation can produce 100 to 200 kg nitrogen per hectare. Wheat is Pakistan main staple food crop. World wheat production is 7,550 million tons. In Pakistan, wheat production 9.12 million hectares, 27 million tons are produced. Total population of Pakistan is now 215.25 million. Population growth rate is 1.80. Fifth most populous country in the world is considered as total nitrogen fertilizer production in Pakistan is 3,925,000 tons. Total nitrogen fertilizer imported in Pakistan is 610,000 tons. Causes of low yield in Pakistan: fertilizer management, poor fertilizer management, nitrogen losses, lack of multiple cropping system, miscropping system, soil salinity due to indigenous use of fertilizer and chemicals, drought spell in rain-fed areas. Rainfall in 2017 in Deccan is only 249 millimeter. Rainfall in 2017-18 Rabi season almost 3 months no rainfall. Monocropping Monocropping is a system in Pakistan in which only one crop is grown in a cultivated area. It has many disadvantages. It is difficult to maintain cover on the soil throughout the year. It encourages pests, diseases, and weeds. It can reduce soil fertility and damage the soil structure. This is a picture of monocropping in Pakistan. Wheat is growing in lines, but you can see here infestation of weeds. These weeds can host for the pest and insect which attack on the wheat and ultimately reduce the yield of wheat. Here, too much wheat present in the wheat field due to monocropping system. Another close picture of wheat during Rabi season, and you can see there are too much weeds in this field. Advantages of intercropping are polyculture. Better use of growth resources including light, nutrient and water, suppressing of weeds, yield stability. If one crop fails due to unforced season, another crop will yield and give income. Successful intercropping gives higher equivalent yield of base crop plus yield of intercrop, higher cropping intensity, reduced pest and disease incidences, improvement of soil health and agroecological system, better control of pests and diseases. Some pests and casual organisms of plant disease are host specific. They attack certain crop species or those belonging to the same family but not others. For example, the problem with rice stem borer will continue if rice is not rotated with other crops of a different family. This is because food will be always available to the pest. However, if legume is planted as the next crop, then corn, beans, and bulbs, even wheat, the build up the pest will be disrupted because they will be deprived of food. Improved soil structure and organic matter due to polyculture or intercropping. The alternate planting of deep and shallow rooted plants will break up the soil. The planting of soybean, other grain legumes, sweet potato and vegetable will return sufficient quantities of plant residue to the soil as their leaves drop on the ground or body parts are left on the field after harvest instead of being burned as is the commonly practiced with sugarcane or wheat or in rice. Green manure will add significant amount of organic matter in intercropping. Improved soil fertility. The continuous growing of single crop will result to the depletion of certain soil nutrients. With crop rotation, soil fertility will be promoted through alternate planting of crops having different nutrient needs. This will prevent the depletion of any one essential element present in the soil. Leguminous plants, because of their ability to accumulate nitrogen by fixing it from the air in association with the rhizobium bacterium, will improve the soil fertility. Rainfall pattern during 2016-17 in Pakistan. Here we see the maximum rainfall occur during the July-August monsoon season, while remaining throughout the year very little rainfall occur or no rainfall, especially in Deccan from October, November, December, and January. These 4 months are totally drought months. October to February usually a drought season in Pakistan, especially in Deccan, which has totally arid conditions. Materials and methods of my experiment methodology: sowing of wheat and intercropping 22nd November 2017. Wheat variety Bakhtavar 92. Row to row distance 0.3 meter. Row length 6 meter. Wheat variety area research area of Department of Agronomy, Global University, DA Khan, Khyber Pakhtunkhwa, Pakistan. Treatment: T1 wheat soil, T2 chickpea soil, T3 lentil soil, T4 one row wheat plus one row chickpea in alternate session, T5 two row wheat plus two row chickpea in alternate fashion, T6 three row wheat and three row chickpea in alternate session, T7 one row wheat plus one row lentil in alternate pattern, T8 two row wheat plus two row lentil in alternate pattern, T9 three row wheat plus three row lentil in alternate pattern. Up now we comes to the results and discussion number of grains per spike. Here we can see that wheat intercropped with lentil produce maximum number of grains per spike. Here the number of grains shows near about 45 or 46 number of grains per spike are produced by lentil one row wheat plus lentil one row lentil. uh, Next, here we see the grain weight or 1000 grain weight of wheat. Here also we see the maximum grain weight was obtained in treatment where wheat one row and lentil one row are in alternate fashion. Here maximum grain weight are 1000 grain weight which are achieved about 45 gram. uh, Number of tillers per plant are also increased by intercropping over sole cropping. Maximum tillers or spikes per plant were observed in treatment where wheat was intercropped with lentil one row in alternate fashion. Remaining treatment remained behind, although we see that intercropping with either chickpea or either lentil proved to be the best over sole cropping. Grain yield ton per hectare. Here we can see wheat and gram, wheat two row and gram two row in alternate fashion, and wheat single row and lentil single row are in alternate fashion. Recorded maximum grain yield about 4.5 tons per hectare, which is which is sufficient in Pakistan, where we obtained 1 or 1.5 ton per hectare in farmer field. Here 4.5 ton per hectare are too much yield which are obtained in polyculture or with intercropped by either wheat with gram or with wheat with lentil in alternate fashion. Grain sheet of all treatments ton per hectare. Here we can see we not only see the yield of chickpea and lentil sole cropping, but also we can see the yield of intercrop and base wheat crop uh, in the figure. Here we obtained additional yields of gram or lentil in addition to the wheat grain yield. Wheat obtained biological nitrogen fixation by these legumes and increase or increment or yield are found by seeing this graph. land equivalent ratio Land equivalent ratio of all the intercropped treatments having more over sole crop because the Addition of legume crop cause more land equivalent ratio than as compared to the sole cropping. Here we can take the advantage of two land equivalent ratio by intercropping legume with wheat crop. Biological nitrogen fixation. Here we can see that from 10 kilogram to 70, 70 kilogram of additional nitrogen fixation in wheat area, which are caused by the legume crops. These chickpea and lentil both are legume crops and cause biological nitrogen fixation in the cell, which is also utilized by the next crop. Maximum biological nitrogen fixation are done by wheat single row and lentil single row in alternate fashion. Although here we can see that lentil are best biological nitrogen fixer than chickpea or grain crops. Lentil fixes more nitrogen in the root zone of wheat in the soil as compared to the chickpea. Pictorial view of the crop. Conclusion: the results revealed that grain yield, number of grains, and seed weight were significantly increased by both intercropping system over sole cropping of each. Maximum wheat grain yield produced 4.5117 ton per hectare in wheat plus lentil one row in alternation intercropping or polyculture treatment, while in sole wheat produced 3.5306 ton per hectare at a minimum scale. Land equivalent ratio was fairly high in all intercropping treatment, but the highest LER was recorded for wheat plus lentil single row intercropping treatment. This is the picture of wheat spikes. Thank you. Now my presentation is over. House is open for the question if you have.
Thank you, Dr. Iktidar, for your presence and presentation. Any questions?
Please, have you any questions, any audience? House is open for question.
Okay, I hope no questions. Once again, thank you for your valuable presentation.
Thank you, ma'am.
Thank you. So by this, we came to the end of day one webinar, and I would like to thank you all attendees and participants of today's webinar. Hope you enjoyed the webinar and gained some knowledge from the sessions, and I request you to attend tomorrow's sessions too. If you have any queries, you can email to the concerned program manager. Thank you.